Very good morning to all the NEET aspirants. This is your Prasanna Ma'am from NEET World, and here we are with our 100 concepts for NEET 2026 session. So today we are going to discuss about those organisms which are not placed in 5 kingdom system of classification by R.H. Whittaker, about lichens, viruses, viroids and prions. And then we are also going to look into the Plantae and animal kingdom in brief. Of course, we learn them in detail in plant kingdom and animal kingdom, but then here we look in briefly about Plantae and Animalia, and then we look into those organisms which are not included in 5 kingdom system of classification. Let's get started. So here if you look into, firstly, Plantae includes what organisms? All eukaryotic chlorophyll containing organisms are placed under what? Plantae. And cell wall of Plantae includes what? Cell wall composition includes cellulose, hemicellulose and pectin. Cellulose, hemicellulose and pectin. Very important beta, you have to remember the composition of the cell wall of plants. This is also given in cell biology under cell wall, also given in plant physiology, also given in plant kingdom. Okay. So cell wall of plant is composed of cellulose, hemicellulose and pectin. And then kingdom Plantae includes 5 divisions. Okay. It includes how many divisions? 5 divisions. First, algae or thallophyta. Okay. Algae or thallophyta, which includes thalloid containing organisms which are photosynthetic and they have autotrophic mode of nutrition. Second will be bryophyta, pteridophyta, third pteridophyta, and fourth is thallophyta, bryo, pterido, gymno and angiosperms. Gymnosperms and angiosperms. These are the 5 divisions under the kingdom Plantae, of which thallophyta, that is algae, bryophyta and pteridophyta are considered as cryptogames. Okay, they are considered as cryptogames. Gymnosperms and angiosperms are phanerogams, which are flowering plants. Phanerogams. Clear right now? Next, alternation of generations. We have 2 phases, okay, in alternation of generations. That one is haplontic or haploid gametophytic phase, and diploid sporophytic phase. And haploid gametophytic phase. Clear right ma? Next, nutrition in kingdom Plantae. What type of nutrition does plants conduct? Holophytic nutrition. Okay, what mode of nutrition beta? Holophytic mode of nutrition. And what is the reserve food material that is stored in plants? Glucose is stored in the form of starch. Clear right ma? And few members of plant kingdom, they show parasitic mode of nutrition also, in which insectivorous plants and parasites are included. We have complete parasites and partial parasites also. Insectivorous plants, best examples include bladderwort and Venus flytrap. Parasitic plants, best examples include Cuscuta, which is a complete parasite. Partial parasite include Striga. Clear right ma? Next, kingdom Animalia. Animalia, if you see, these are heterotrophic, eukaryotic, multicellular organism. There is no cell wall in the cells of animals. The limiting boundary of the cell is cell membrane. Cell membrane is the outermost limiting boundary of plant cells. Nutrition is strictly holozoic mode of nutrition. Reserve food material includes glycogen. Glycogen is the reserve food material in animals. they show definite growth pattern and locomotion, whereas plants will show indeterminate growth, where they grow throughout their lifespan, but then animals will show definite pattern of growth, and they exhibit elaborate sensory and neuromotor mechanism. When it comes to reproduction, they show sexual reproduction followed by embryological development. Okay, this is a brief about kingdom Plantae and Animalia. Now let us look into virus, viroids, prions and lichens, which are not placed in Whittaker 5 kingdom system of classification, not placed in Whittaker's 5 kingdom system of classification. Clear, right? Now you see it clearly, ma. They are not included in 5 kingdom system of classification. Let us learn one by one. Okay, viruses. Viruses are the infectious agents. Viruses are not called as non-living organism. At the same time, we can't call them as living organisms as well. They are well described as living entities or crystalline structures. Okay, they are well explained as what, beta? Crystalline structures or living entities, and these are infectious agents. They are non-cellular organisms. So viruses are exceptions to cell theory as well, because they don't have cell. Okay, cell contains what, ma? Limiting boundary, that is cell membrane. Protoplasm should be there, that is cytoplasm and nucleus. As virus doesn't contain a proper cell, so cell theory is not acceptable or applicable to virus. This is an exception to cell theory as well. And these are connecting links between non-living entities and living things. Okay, living and non-living entities, viruses are the connecting links. Outside a living cell, viruses are considered just as crystalline structures. They are just considered as crystalline structures outside the living cell. Inside the host or living cell, they are assumed as they conduct the replication process and they increase in the number. They conduct the replication and increase in number inside the living or host cell. Clear with these aspects, beta? Next. Here if you see discovery of viruses, how virus is being discovered. So if you see, if you look over here, firstly DJ Ivanovsky in the year 1892, what did he say virus as? Poisonous fluid or venom. Virus he considered as poisonous fluid or venom, wherein he said that viruses are the smaller structures than bacteria. They can easily pass through bacterial filter paper. Okay, they can easily pass through bacterial filter paper. Okay, he said that virus is a poisonous fluid or venom which will cause the disease in the tobacco mosaic plant by the virus tobacco mosaic virus. He didn't named it yet, but then he has considered it as what? Poisonous fluid or venom which cause infection to the healthy plants, and the size of the viruses will be smaller than bacteria. They can easily pass through bacterial filter paper. Continuation of his experiments are conducted by W.M. Beijerinck in the year 1898. He said that this poisonous fluid, when it is transmitted to a healthy plant, this also get infectious. Okay, and this phenomena he has explained it under contagium vivum fluidum. Fine. So the fluid which is extracted from diseased plant, when that is transmitted to a healthy plant, even that gets affected. So this he has explained under contagium vivum fluidum, and it is the scientist W.M. Stanley in the year 1935. He has crystallized virus. crystallized virus particles. He has crystallized virus particles and said that virus are nothing but the crystalline structures outside the living host. So these three scientists and their points are very important for us beta. First is DJ Ivanovsky, who said or considered virus as a poisonous fluid or venom. He said that virus structure is smaller than the bacteria and they can easily pass through bacterial filter paper. Second is W.M. Bajering, who said that this poisonous fluid, when transmitted to a healthy plant, that also gets infectious, and he named the concept as contagium vivum fluidum. W.M. Stanley, in the year 1935, he has crystallized the virus particles. Clear with this, right? Now let us look into the virus types also. Most of the viruses will just have a genetic material and a protein coat over it. That protein coat is considered as capsid. But then there are few viruses, over the capsid also there will be one envelope, which are said to be enveloped viruses. So here what are the three constituents of viruses? Virus can have envelope, a proteinaceous coat called as capsid, and then a genetic material. Genetic material can be either DNA or RNA, but not both. Okay, it can be either DNA or RNA, but it cannot be both. So here envelope around the virus, so they are called as what? Enveloped virus. Best examples for the enveloped virus includes HIV, herpes virus, influenza virus. Okay, so these coronavirus, these are the best examples for enveloped viruses. Okay, next capsid. Capsid will contain protein subunits called as capsomeres. Capsid will contain what, ma? Protein subunits. Protein layer is capsid and they have protein subunits called as capsomeres. They are arranged either in helical or polyhedral or geometric forms. Okay, so this is about envelope and capsid. Next question, next slide. So here, good afternoon, ma. Continue with the class. Any doubt anywhere, you can just drop a message in chat box. I will be explaining. Your name is Abdullah, right? Yeah. Continue with nucleoid. So we have discussed about envelope and capsid. Now nucleoid. Nucleoid can either be RNA or DNA. Animal infecting viruses. Okay, animal infecting viruses will definitely have double-stranded DNA. Okay, animal infecting viruses will have what genetic material with a double-stranded DNA. Best examples include pox virus or herpes virus. Second is plant infecting viruses. Mostly they have what genetic material? Single-stranded RNA, which includes TMV or HIV. Next, bacterial viruses or bacteriophages. Bacteriophage or bacterial virus, they also have double-stranded circular DNA. Okay, they also have double-stranded circular DNA. This is clear to you, right? Animal infecting viruses will have double-stranded DNA. Best examples, pox virus and herpes virus. Plant infecting viruses, mostly their genetic material will be single-stranded RNA, TMV and HIV. Next, not HIV, tobacco mosaic virus, or you can also take cauliflower mosaic virus. Okay, they have single-stranded RNA. Bacterial viruses or bacteriophages, they will be having double-stranded DNA. Clear, right, ma? Next. Here you see viral infections. Viral infections can occur to animals and plants. In animals, viral infections, few viral infections include mumps, chickenpox, herpes, influenza and AIDS. Here in plants, tobacco mosaic disease is caused by tobacco mosaic viruses. Okay, Cauliflower mosaic disease is caused by cauliflower mosaic viruses. Clear, right ma? Next. Symptoms of viral infections in plants. What will be the symptoms of viral infections in plants? First, mosaic formation, then leaf rolling like this, okay, leaf rolling takes place, leaf curling and yellowing of leaf, vein clearing, okay, the veins which are present, okay, mid-rib or the lateral veins, they will get cleared, okay, vein clearing. Next, dwarfing or the stunted growth of the plant will be occurred. So, these are the symptoms of viral infections. Make a key note of this and remember all the symptoms of viral infections. We can expect a question from here, okay. You should not get confused if some other symptoms are given and asked you the combination of symptoms for viral infections. These are the symptoms. You can remember that. Very easy, right ma? Next. About tobacco mosaic virus. Many a times we got question on tobacco mosaic virus ma. Let's learn it in detailed way, okay. First, this is first observed by whom? DJ Ivanovsky, okay, as poisonous fluid or venom. Next, what is its structural description? This is described as an elongated rod-like structure with how much angstroms long? 3000 angstroms long. So, this is an elongated rod-like structure like this, elongated rod-like structure with 3000 angstroms. And what will be the diameter? Diameter is 180 angstroms is the diameter. What is the molecular weight of it? 39.4 into 10 to the power of 6 Dalton's molecular weight, okay. And as we have discussed, it is encovered by a protein layer called as capsid, that is having protein subunits called what? Capsomeres. There are around 2130 capsomeres and these are the capsomeres. This is complete protein coat. Around how many capsomeres are there? 2130 capsomeres. And as it is infecting plant, right, tobacco mosaic virus, it has RNA, single stranded RNA. This single stranded RNA will have 6400 nucleotides approximately. So, how many nucleotides does it have ma? 6400 nucleotides. So, now let us look into the ratio between nucleotides and capsomeres. The ratio is 3:1, okay, 3:1 is the ratio. Between the number of nucleotides and capsomeres, the ratio is 3:1. Clear, right? Next question. Next, if we look into the bacteriophage, TMV is clear to you now? How many capsomeres we have? 2130. How many nucleotides in the RNA? We have 6400 nucleotides. If we have to take the ratio, that is 3:1. Bacteriophage, you see, bacteriophage, it is infecting bacteria. So, what is the genetic material? It has double stranded DNA. What is its structural description? It has tadpole structure with polyhedral head and a helical tail, okay, it has polyhedral head and helical tail. This head will have double stranded circular DNA, it has neck and tail, neck is connected to the tail, neck is connecting head and tail, clear, right? And tail will be having tail fibers, tail will be having what? Tail fibers. This tail fibers at the end, they have enzyme called as lysozyme. Tail fibers will end with the enzyme lysozyme to lyse the cell wall of the bacteria. Okay, when this comes and attaches over the bacteria to lyse the cell wall of the bacteria, the tail fibers will be having what? Lysozyme. Clear with the description of bacteriophage? Next, about viroids. Viroids are first described by Theo Dionner in the year 1971 by Theo Dionner. These are viruses without protein coats. Viruses with their coats off are viroids. They do not have protein coats, they just have the genetic material. Okay, they just have the genetic material, that is mostly RNA without protein coat. They cause what disease? Potato spindle tuber disease. They cause potato spindle tuber disease, and mostly they have what RNA? Single stranded RNA which tightly folds to form circular or linear structures. This single stranded RNA will tightly fold and form circular or linear structures like this, okay, and they have low molecular weight. They have very low molecular weight when compared to viruses. So, clear with viroids ma? Viroids, they just have the genetic material and it is RNA. They are causing what disease? Potato spindle tuber disease, and they do not have protein coat, it is discovered by T.O. Diner. Their molecular weight is less than that of viruses. Next, prions. Prions are nothing but abnormally folded proteins. Unlike viroids, viroids will have just genetic material, that is RNA, whereas prions are having abnormally folded proteins. They in them nucleic acid is absent. In viroids, protein coat is absent, in prions nucleic acid is absent. So, it has what? Abnormally folded proteins. They cause neurological disorders. Examples include scrape disease in sheep, mad cow disease in human, mad cow disease in cattle. Okay. So, there is one more name for this mad cow disease in cattle. It is by bovine spongiform encephalopathy. Okay. Bovine spongiform encephalopathy. Bovine spongiform encephalopathy. Clear? And so, this an analogous variant of this mad cow disease is CR-Jacob disease, that you can shortly call it as CJD in humans. So, these disease names are very important, and here I will give you one example. See, the analogous variant of bovine prion protein, and same analogous variant you can see in the humans also, same type of variant. Okay. So, with respect to prions, they are nothing but abnormally folded proteins, nucleic acid is absent, they cause neurological disorders. Best examples, scrape disease, mad cow disease by bovine spongiform encephalopathy. This is also called as BSE. Analogous variant in humans is CR-Jacob disease, and this is the analogous structure that I can show you. Next, molecular weight, if we have to look into prion molecular weight will be less than viroid, it is less than viruses. This is the molecular weight in ascending order: prions, viroids and viruses. In descending order: virus, viroids and prions. Remember that. Next, about lichens. Okay, lichens. Lichens are nothing but the symbiotic association between algae and fungi are called as lichens. These are the pioneers of plant succession. So, in them algal component is called as phycobiont, fungal component is called as mycobiont. So, this algal component will prepare food and they provide nutrition to the fungal component. Fungal component will provide shelter to the algal component. Amongst algae and fungi, which is bulk, fungal component is bulk. Okay. So, this is about lichens. And next, lastly, so here can you see algal and fungal component? This is the algal component and this will be the fungal component. Just 5% of the total lichen will be having algal component. They are very good pollution indicators. How they are very good pollution indicators? I will tell you, this is a very fascinating thing actually. We know algae is autotrophic, conducting photosynthesis. Photosynthesis is possible only through the pigment chlorophyll. We know the structure of chlorophyll will have what porphyrin ring and phytol chain. Yes or no? So, it has porphyrin ring and phytol chain. At the center, magnesium will be present in the porphyrin ring and then phytol chain. So, when this is exposed to the pollution like sulfur dioxide or nitrates, what happens is the bonds between this will be cleaved and this forms magnesium sulfate or magnesium nitrate. Fine. magnesium sulfate, magnesium nitrate, like that bonds will be formed, whereas the bonds will be breaked in the chlorophyll structure. When the structure of chlorophyll is diminished, is there a possibility of photosynthesis? No. As such, no photosynthesis, no nutrition, fungi cannot survive. So, what happens? The lichen cannot survive in the pollution areas because it is very much sensitive to the pollution or pollutants like sulfates and nitrates, mostly for sulfur dioxide type of pollution only. So, that is how they are called as good pollution indicators. They do not grow in polluted areas. So, with this we have completed the entire chapter of biological classification. In our next class, we will be starting plant kingdom, from which we can expect again 3 to 4 questions for sure in the final NEET examination. Definitely 2 to 3 questions will be appeared from plant kingdom. Okay, connecting to that like ploidy and the phases, bryophyta, pteridophyta, gymno, angio, alteration of generations, examples, everything we are going to discuss in a most detailed way. Okay, the only thing you have to do from your end is spend just half an hour and listen to the class, so that each day the preparation goes on with consistency. Okay, so stay tuned with NEET World to know more and to practice more for NEET 2026, and thank you.